Recently, scientists from around the world have begun to synthetically create clones of various organisms with a wide range of success. One story of success is that of Dolly the sheep, the first mammal cloned from an adult somatic cell. During the first step in the cloning process, scientists extracted a cell taken from the udder of a 6-year-old Finn Dorset sheep. Then they took an immature egg cell from a Scottish blackface ewe and removed its nucleus. To remove the nucleus, scientists had to use a small suction pipette to hold the egg steady and a glass needle to poke through the membrane and carefully remove the nucleus. The extraction is monitored closely under a microscope to ensure that no errors are made that would harm the rest of the egg. After that, they insert the donor nucleus from the mammary cell into the egg cell and fuse the two with a jolt of electricity. The electricity also helps to kickstart the cell into activity and make it more likely to divide. The fused cell was then placed into a surrogate sheep of the same breed as the egg donor. Later, the surrogate sheep would give birth to a lamb that had the same genetic identity as its DNA donor. This lamb would be called Dolly, a name given to it because of the cell its DNA was taken from. Because of the electricity, the sheep is born with a lightning scar on its forehead and becomes a wizard. Scientists originally believed that only the genes in the fertilized egg, embryo, or very young animal are fully activated and capable of creating an entire organism. However, Dolly proved to scientists that the DNA in the nucleus of an adult somatic cell had the same information needed to create the entire sheep and could be reprogrammed to do it. Although Dolly died at the young age of 6 from lung disease and suffered premature arthritis, she was considered the first successful mammal ever to be cloned from an adult somatic cell. Whether the fact that she was a clone had any significance in her early death is a mystery, but whatever the reason, Dolly is still celebrated as the first of her kind.